Welcome to this episode of Eureka, and this is a COVID special Eureka. We are talking with number of scientific institutions and people who are heading them to see behind the screen how Indian science is helping the government of India in fighting this pandemic. The frontline workers are, of course, the medical professionals, the administrators, and whatnot. But if the war has to be won, the science and technology is important. And these are people who are behind the screen, who are pushing the frontiers, trying to understand the virus, find solutions, reduce the rate of death, find innovative ways to arrest this pandemic. And in today's discussion, we are going to have Dr. Ram Vishwakarma. He is the director of Indian Institute of Integrative Medicine, an institution in Jammu. It is an institution part of CSIR. And thank you, sir, for being in the show. It's a very wonderful opportunity. Thank you for being here. So let me start with the name of your institution itself, because many of us may not be even able to get an idea. It says that integrative medicine. So what does it mean? It's a different terminology that many of us are not familiar with.
Yeah, so integrative medicine means bringing ideas of modern science, tools and concepts and technologies, and looking at the concepts, materials, plants of traditional medicine. So that's one pathway. The other pathway is doing the reverse, using traditional medicine, their concepts, and then validating with the modern clinical pharmacology, modern physical sciences, and taking them all the way to clinical trials and validate.
Okay.
So it is new to most of the people, but if you go into history, the modern medicine actually started 200 years ago with deep roots in traditional medicine in the world.
Okay.
It started with the isolation of first pure molecule from a medicinal plant called morphine. All of us know 200 years exactly. We celebrated 200 years recently.
Yeah.
Turner's discovery of morphine. And if you go to all FDA approved drugs, almost 50% drugs you will find they have come from different traditional system of medicine ideas or plant material. So we decided renaming this institution in 2007 during Dr. Mashelkar's time. That time has come that we have to bring these two traditional knowledge and modern science together so that benefits the traditional medicine and benefit the modern science and modern medicine.
Very, very interesting. I mean, the effort is revitalization of medical practices that was practiced not just in one part of India, but different parts of India, maybe world over, and bring it together, give it a modern evidence-based sound ground so that it can be taken forward, right? That's a very interesting point, I suppose. Today, when we are facing quite a lot of challenges, when we need to provide a solution to a large number of people, maybe we have to think out of box. One of the areas that you are also working is looking at natural products, using natural products as medicine, and there is also a technical term that you use, that phytopharmaceutical, right? So what does this phytopharmaceutical actually mean? How does that word come in?
Yeah, so I will explain this to you in regulatory term that will make it very clear.
Yeah.
So traditionally, plant and microbe have been primary source of new medicine.
Okay.
So like today, all the anti-cancer drug that you use in modern clinics, starting from Taxol, paclitaxel, most of them have come from plant sources.
Okay.
Most of the antibiotic that you use today in modern medicine, almost all have come from microbial sources.
Yeah. Okay.
All these mycins, erythromycin, they all come from bacterial sources. So
even the first discovery of antibiotics is from a microbe, right? Like
microbe, exactly, penicillin, right? So natural products have been the primary source of discovery, and it is not very surprising because if you look from evolutionary point of view, plant life and animal life have evolved together, and life had existed on for thousands of years. We have been there, okay, and modern medicine has been there only for last say 100 years and 200 years. So traditionally, all over the world, communities have been using plant around them, microbe around them, fungus around them for their medical need. And much of it has been documented in the form of, say, Charak Samhita and other Chinese traditional say, medicine system. So, nutshell, natural products have been primary source of drug for modern medicine. Now, these natural products are a small molecule natural product isolated from plant, fungus, algae, or bacteria. Okay. And this institution where I work, the center of gravity of this institution is those natural products. So we study them in all dimensions. We study them, how to isolate them, how to determine their structure, how to synthesize them in the lab. So we have major synthetic chemistry program, and how to modify them to convert them into medicines, and then how to study their biosynthetic pathway within the plant system, how plant make them.
Okay.
And going next stage further, why plant make them. So all these questions we are addressing in this laboratory, and we are very fortunate that we have two campuses, one in Jammu, one in Srinagar, and 6 experimental stations around the J&K at different altitude in Himalayas. From Ladakh to Kashmir, we have those stations. Around 300, 400 acre of land is available to CSIR. Okay. And we cultivate all the medicinal plant of Himalayas are available to us. We grow them, we develop agro-technology, we transfer technology to industry, and that is how we work. Now we're coming to regulatory term that you asked. So primary source, people have been isolating these pure molecule and developing them as a drug, what we call NCE. Okay. That is well-tested pathway that we have been following. Now, this need was felt for many years that whenever you isolate a plant, a compound from a plant, invariably compound may start losing activity. Okay. Or start developing resistance. So I give you one example. There is a famous Chinese plant on which a Nobel Prize was given, plant called Artemisia annua. Yeah. From which this artemisinin was isolated. And a lady from China got a Nobel Prize few years ago. Yeah. Now this plant has been used as a concoction in China for 5,000 years. Okay. And it's been continuously working. But this molecule was isolated in 1960s. It got developed as a FDA-approved drug in 1980s, and has been used for last 40, 50 years. But we are now seeing that resistance is developing against this particular molecule in malaria parasite. Okay. Okay. It brings down the question that why this is happening. So the scientifically we are looking at perhaps when you have several molecules in the plant, they are not allowing microorganism to develop resistance against this molecule. Okay. So this concept came that why we are isolating these compounds, if we can develop a particular well-defined fraction which contains well-defined compounds, develop as a drug. Okay. As a modern drug, not as a drug. And this pathway was opened by US FDA in 2005. Oh, I see. They named it Botanical drug. Oh. A separate category of drug was added by US FDA for modern medicine called Botanical drug. I see. Okay. And since then, 3 drugs have been approved by that agency. So in India also, we started working with Drug Controller General that India should also have this pathway. And finally, we succeeded in 2015. To this notification, Government of India passed this Gazette notification, and DCGI then promulgated it, that now we will start approving phytopharmaceutical drug. So now in our institution, we have, since we are plant-based, natural product-based institution, we took lead in this aspect. Okay. And we are developing several medicine, for example, for arthritis, for Alzheimer, basically diseases which are lifestyle related or diseases which are chronic. Okay, so basically one of the aim is how to address some of these chronic diseases for which, for example, there is no magic bullet kind of stuff.
Like if you are suffering from diabetics, it's not like one pill is going to cure you.
Exactly.
Where you may have to do quite a lot of. So let me ask you specific questions. I mean, can you illustrate some 2 or 3, let's say, areas that you are working on, 2 or 3 diseases that you are working on, where you are trying to develop this botanical-based drugs? Yeah.
So one area that we are working on is area, like I said, the area of chronic disease, for example, rheumatoid arthritis and arthritis.
Okay.
Okay, these are basically rheumatoid arthritis is an autoimmune disease, okay? And arthritis is basically bone degeneration disease,
Yeah.
and related to aging process and deficiency of certain minerals, certain materials.
Yeah.
And of late, even people are getting these diseases at the early age.
Yeah.
Okay. So, being multifactorial disease where several proteins and receptors are involved in the etiology of disease, we have identified certain plant material, and one of them is actually is going for clinical phase 1 trial. In last week, we had presented to DCGI and hoping that we will get approval and that will start. And
Okay.
what that plant, and we know the molecule also, what is doing it, it's a very potent IL-6 inhibitor. IL-6 is a pro-inflammatory cytokine,
Yeah.
which is causing inflammation and causing arthritis. And these, so this is one area, arthritis and rheumatoid arthritis.
Yeah, so let me ask you one question there. I mean, let's say, for example, you said that you are developed from a particular plant. Maybe you don't want to name the plant, that's okay. But how did you zero in on that plant? I mean, there are billions of plants around. So how do you know that this plant might have a, that's a great mystery.
Yeah. So we follow two pathways. One, we use our traditional knowledge. If, for example, plants, X number of plants are listed in traditional knowledge of, say, Indian medicine or any international medicine system, traditional medicine. So that is one clue you can get. If people have been using a particular plant for hundreds of years, there must be some wisdom in that, and they are documented. So we take that plant, grow them in our farm, and test them and see whether that claim is right or wrong. That's number one. Second is because of the lot of information in terms of the protein structure databases. There are now millions of proteins available to us, and there are many of thousands of X-ray structures are available. So we do in silico effort. We do large scale millions of molecules we can screen and then identify the best fit. So what we are doing at the institute, we are maintaining a natural product library.
I see.
Pure natural product library, and we do in silico effort. And from that, then we select and then go back to the plant.
I see. Okay. So which plant produces this? That's how you go back.
So this will give you integrated medicine idea. The one idea is coming from traditional medicine. Other idea is coming from modern tools of science and crystallography, NMR spectroscopy, genomics, and all the structures, and then identifying the ligands. So both these approaches are followed.
No, see, you said that we are now ready for going to clinical trial 1. So, but before clinical trial 1, what do you do? How do you assess that it is mature enough to test in the humans?
So there is a long process. A new drug discovery is a long process. All of us understand. What we do, first we identify what we call a hit molecule. And we have a certain definition of it, that what qualify a hit means engagement with the biological target. It could be an enzyme, it could be a receptor, it could be ion channel, number one. Then we identify and set up a appropriate animal model, okay? A mice model, a rabbit model, or whichever model, and test an in vivo model. Then we have to do what we call pharmacokinetics, how this molecule goes into our body, how much it goes in the blood, how it circulates throughout the body, that knowledge we generate. Then next step comes, we do the regulatory safety. Whether it is safe, 28 days and 90 days safety. Then safety data comes, then we go to GMP manufacturing. So we have luckily GMP manufacturing plant in the institute. The only plant in India is available in our institute.
I see.
For phytopharmaceutical drug, which was funded by DST, CSIR, Ayush Ministry. 3 people funded this, and this is up to us. So then we make these clinical trial batches. We compile all this data. We go to the drug controller, and then for approval, and then we collaborate with hospital and do the clinical trial. That's a pathway.
Okay, so it means that quite a bit of research work goes on. I mean, from identifying the plant or identifying a potential chemical, trying out and so many things before you come. It's a very interesting thing. We'll take a very short break here. After the break, we'll continue this conversation. Keep watching Eureka. We'll take a very short break. After the break, we'll continue this conversation.
Khadi masks manufactured by Khadi and Village Industries Commission are helping people fight COVID-19 pandemic and keeping the infection at bay. Till now, over 7.5 lakh Khadi masks have been distributed to the district authorities by the Khadi institutions across the country.
Welcome back to Eureka. We are having a very wonderful, very enlightening conversation with Dr. Ram Vishwakarma. He is the director of Indian Institute of Integrative Medicine, a constituent unit of CSIR, and they are working on very exciting projects of how to harness plants and microbes to produce drugs and pharmaceuticals to save us from various diseases. Before the break, we are talking about what is this integrative medicine, how they are finding drugs from hundreds of plants and microbes, and we had a very wonderful explanation from him. So, sir, continuing that, can you give us some more example of the work that you are right now doing? You said that one Botanicals is going to go to clinical phase trial 1, so which means it's a really good matured level, good potential. Of course, one needs to see what happens in this clinical trial. As a scientist, one would always take a skeptical position. That's true. But still, going up to this level is a great stuff. So any other example that you can say that would be very interesting for us?
Yeah. So second example I will give is that we have been working on this very important plant from Western Ghats, a plant called Disoxylum binactiferum. This is a very important plant, and it is not listed in any traditional system of medicine.
I see.
So we don't restrict ourselves to the traditional system. We focus on biodiversity of India.
Okay.
So we started working on this plant and found a very interesting property, what we call anti-inflammatory property.
I see.
But later on, when we tested this under cancer cell lines, very potent activity against pancreatic cancer and certain blood cancer cell lines.
I see.
Okay. So when our journey started almost now 9 years, and we isolated pure compound called rohitikine, which had all the activity that I described. The only problem with this molecule was that it was not bioavailable. When I say it was not bioavailable means when you give it to animal model, very little amount, if you give 100 milligram, only few microgram end up in the blood. So that means it is not fully absorbed. So then we did a project how to improve bioavailability, and it took us 2 to 3 years. And then we ended up this molecule which we have named for IIIM 290. Okay, this is a pure molecule drug, and we have done all the work, including xenograft model on animals, safety, everything. And this we are taking it for clinical phase 2 trial for pancreatic cancer, for which there is currently no drug available. So this is, I consider, as a breakthrough of our inquiry, which took us almost 8-9 years, and this very soon you will hear about it for pancreatic cancer.
Very, very nice. I mean, you say that it means that clinical phase trial 1, we have had a good success. That's what is resulted so?
Yes, phase 1 / phase 2, because cancer you have to do phase 1 because cancer trials are done on the patient itself.
You
cannot do healthy volunteer. So we are doing phase 1 / phase 2, where we are looking at the safety and efficacy simultaneously. Normally in phase 1 trial, you look at safety, and phase 2 trial, you look at efficacy.
So now in the cancer patient, you won't get so many numbers and you can't do it in a normal person. So you may have to do it.
Ethically, anti-cancer drugs are not allowed to be tested on normal healthy volunteer.
Okay.
Yeah. So you have to do safety also in patient and the efficacy also in patient.
So where will this trial be taking place, in India or outside or in different places?
Trial site we have identified one in Bangalore. and one in Medanta Hospital here in NCR region.
Okay.
So these two trial sites will happen on pancreatic cancer, and this is the most deadly cancer right now, where we do not have any cure. And I am really keeping my fingers crossed that this vital plant from Western Ghats will provide us this medicine. And we filed a US patent for this, and we have published this work also in Journal of Medicinal Chemistry of American Chemical Society. So this is something we take very feel proud about this.
Yeah, very nice, very nice. Sir, now let me move slightly forward and then talk about the current, problem that we are facing, which is the COVID-19 pandemic. How is your institute gearing itself to helping the efforts in arresting the pandemic?
Yeah. So we are doing 4 things. First, since we have a BSL-3 lab in our institute, we are helping Jammu and Kashmir government to do testing of COVID patients.
Very nice.
That's one. Second, we have started a clinical trial on a product known as Sepsivac.
Okay.
Which is a basically heat inactivated Mycobacterium W. And this is a product of Cadila Pharmaceutical and CSIR collaboration. Long time collaboration that this company from 2007 onward. And Sepsivac is already approved as a drug for gram-negative sepsis.
Okay.
Sepsis caused by gram-negative bacteria. And incidentally, the pathology of gram-negative sepsis and the viral sepsis by COVID-19 are very similar.
So when the viral infection takes place, I mean, when the novel coronavirus infects you, most of people recover on their own. I mean, nothing much needs to be done. But when they get into critical problem, actually the problem is actually sepsis, right? I mean, because of bacterial infection, the sepsis spread through your blood to different parts of your body and things like that, right?
Yeah. So this drug, basically, we are right now doing phase 2 trial in All India Institute of Medical Sciences, Delhi, PGI Chandigarh, and All India Institute of Medical Sciences, Bhopal. So this trial is about to complete in next week. And then we will do 2 phase 3 trial on 1100 patients, and the phase 3 trial.
Okay. So which means that the phase 2 most likely is going to be ending very soon, and you're going to start phase 3. And this drug, because it's already approved a drug, so then it's not much of a difficulty in talking about its safety, etc. Right? I mean, so that is solved.
Yeah, it is approved, and it works through boosting the Th1 component of innate immunity.
Okay.
So, which is like BCG, half of the BCG. BCG has Th1, Th2 both. This Mycobacterium W has Th1. So, we have very solid hypothesis, and we hope that we'll get the result which will help the critical care patient in the ICU situation.
I see. Okay, that's a interesting point. So this is the second one. You said that you are working on 4. One is using your BSL-3 lab, and second is this Sepsivac, and then
third is we are working on a very interesting plant. We have been working for last 5 years on a plant from Madhya Pradesh and Chhattisgarh tribal belt. This plant we were working for. Dengue virus. As you know, dengue virus is a positive RNA virus, very similar to this virus. The difference is that this virus uses a different route to enter the cell, and dengue uses different route to enter the cell.
Yeah.
Once they are inside the cell, the endocytosis mechanism is more or less similar.
You mean to say that because both are RNA virus, what they do inside the cell is similar? The way they may enter might be different. This may be using AC2, and then that may be using something else. But once inside, it's the same. The ribosomes makes copies, and then Golgi makes it into package, produces new virus. So where is this antiviral is going to work? I mean, is it going to work as what? I mean, how exactly is that mechanism happening?
So this will block the viral post-translational glycosylation processes.
Okay.
Number one.
Uh-huh.
It will block the endosome mechanism of virus entry. But after entry, virus maturation going to ribosome. So we have tested this in animal model, and fortunately we had completed phase 1 trial for COVID. So safety data is also there.
Okay.
This is in collaboration with ICGB New Delhi and a major pharmaceutical company of India who will conduct the trial.
Okay.
And happy to tell you, last week we had received phase 2 trial approval for ACQH.
Oh, I see.
And maybe this week we are going to start the trial.
I see. Okay. Okay. So this also is going to take place in number of hospitals around the country.
Absolutely. Absolutely. So this is also going to be a very important product, which I'm hoping. And good thing about MW and ACQH are these are totally Indian discoveries. These are the demonstration of capabilities of Indian science.
Okay.
And collaborative ability of multiple companies and institutions to work together and come to this stage.
And also, it's very interesting. I mean, actually, this botanical ACQH, you are not doing it for COVID, but you are doing for something else. But because the mechanism is similar, you can also repurpose it for this sudden emergence, right? I mean, that's a very interesting.
I consider myself fortunate that we had phase 1 data and we could do. Otherwise, we could have not been able to do this.
Yeah, it will not be that. So there is a fourth component that you talked about, right? One is testing, the second is that wax, and then this botanical, and fourth one?
Fourth component is where we have signed, you must be getting in the newspaper, the Ayush Ministry and CSR has signed a very comprehensive agreement.
Okay.
Where we have identified 4 Ayurvedic plants used in Ayurveda. Okay. And we will conduct large-scale trials on these 4 drugs.
Okay.
For COVID-19.
I see.
And this effort will be executed by the IIIM, my institute, on behalf of CSIR.
Nice.
This is first time maybe in India when these Ayurvedic plants are being taken for modern trials. These will be done in modern hospitals, the way we are doing other trials, and we create a scientific evidence for these particular drugs. And China recently, in the last 2-3 months, has very efficiently used their medicinal plant knowledge to deal with the problem of COVID-19.
And by using evidence-based science, that's a key component. That's very interesting. I mean, one can see that how by mere existence of such an institute and human resources over there, when a challenge comes, that we are able to gear ourselves up. So when this institute, I was told that this institute was formed way back in 1941, even before India became independent. Can you tell us as a snapshot the history? I mean, you should have a very long, rich history, but some glimpses of it, please.
Yeah, so this institution was set up by a very major figure of Indian science, Sir Ram Vishwakarma, who was father of Indian pharmacology.
Okay.
And he set up this institution in 1941 when J&K was an independent country.
Okay.
And in those days, it used to be called Drug Research Laboratory of J&K.
I see.
And then then after the independence, this became part of CSIR. And its name became Regional Research Laboratory.
Okay.
Like other regional research laboratories, Hyderabad and others. And in 2007, during Dr. Vishwakarma's period, because of the reason I gave you, it was focused on this particular niche of science, modern medicine, and traditional medicine. This institution was renamed as Indian Institute of Integrated Medicine.
I see.
A very proud legacy of Sir Ramnath Chopra, who was the one who wrote the Indian Drug and Cosmetic Act of India in immediately after independence.
I see. It's a very nice history that you have, and then also looking at many of your products, at least 3 you said, 4 actually you said are going for human trial. It's a huge thing. I mean, not many labs actually have 4 drugs which are going for human trial. I think it's not only a legacy that one would look back, but also a heritage that we will look forward to. Yeah. That's nice. Thank you for being in this show. It was a very wonderful conversation with you. Thank you for being here. Thank you. Keep
My
watching
pleasure.
Eureka. We'll come back with a interesting conversation next week, same time.
Thank you.